In this video, we are going to focus on the study of pharmacodynamics and toxicodynamics and the role of receptors and ligands. The learning objectives for this lesson are to become familiar with the overall concepts of pharmacodynamics and toxicodynamics, identify different types of ligands, and identify different types of receptors. We have covered what the body does to a drug or toxicant by studying ADME, the absorption, distribution, metabolism, and excretion of a drug or chemical. Once a drug has been administered, it gets absorbed into the systemic circulation, travels through the bloodstream, and exits the bloodstream, and finally reaches its target site. This next section will cover pharmacodynamics, or what the drug does to the body. This requires zooming in to the target site to investigate what happens at the target site, and then stepping back to understand how the target cells alter a whole organ's response and how an organ response results in a whole body response. To get an idea of the pharmacodynamic process, let's look at a cell. Here we have the outline of a cell with its nucleus containing DNA. Two key players in pharmacodynamics are the ligands and receptors. Ligands bind receptors and can cause structural changes and/or recruit additional proteins to the receptors to induce signaling pathways. These signaling pathways use second messengers such as cyclic AMP and calcium signaling, which can be integrated within the cell to propagate a specific signal to the nucleus, triggering changes in transcription levels. As a result, a change in protein composition of the cell may occur, creating a cellular response that is then continued beyond the cell through cell-to-cell communication, resulting in a tissue and whole body response. Let's briefly go over the key elements of pharmacodynamics that we will discuss in more detail in this part of the course. Ligands are chemicals that can be naturally occurring, such as neurotransmitters, drugs, such as aspirin, or toxicants, such as lead or the toxins in the plant oleander. These ligands can move throughout the bloodstream and reach the target site where they bind receptors. Receptors are commonly located on the cell membrane, such as G protein-coupled receptors, receptor tyrosine kinases, and ion channels. Membrane-bound receptors bind hydrophilic ligands that cannot easily cross the membrane. Receptors can also be located in the cytosol, where they typically bind hydrophobic ligands, such as hormones. Keep this in mind as it adds a level of regulation and control to how drugs and toxicants can induce a response. After receptor binding, a cellular signal is triggered and amplified throughout the body, resulting in a response that can be measured. You can quantify a response using dose-response curves, measuring an individual response, or evaluating a whole population's response. Ligands can be almost anything. Here is a short list and some examples to get you started thinking about which compounds can be ligands. Ligands can be peptides, modified amino acids, chemical elements, glycosylated proteins, steroids, pesticides, toxins, drugs, and more. There are many ways to categorize ligands, but one of the most common ways is based on the type of response they trigger. When agonists bind a receptor, they trigger a full physiological response. Endogenous ligands in the body are full agonists. Endogenous ligands, like epinephrine, insulin, or acetylcholine, bind their respective receptors and trigger a full response. Partial agonists result in less than a full response. An example would be buprenorphine, which even at high concentrations will never trigger a full response at the mu-opioid receptor like morphine will. Inverse agonists are ligands that when bound decrease a response below the basal level of activity of a receptor. An inverse agonist has all the properties of a full agonist except that it elicits the opposite response of a normal agonist, or in other words, it demonstrates negative efficacy. As the common definition of an inverse agonist became clear, more and more molecules that exhibit inverse agonism with clinical application have been established. Antagonists bind to receptors without activating them. There are two types of antagonists: competitive and non-competitive antagonism. Competitive antagonists block the effect of an agonist in a reversible way, and the action can be overcome by increasing the dose of the agonist. In other words, the block is surmountable. The action of non-competitive antagonists cannot be overcome by increasing the dose of agonist. This is because the agonist and antagonist bind sites that are different. Hence, the agonist will not displace the antagonist molecule. For example, ketamine binds to the NMDA receptor channel pore, but the agonist, glutamate, binds to the extracellular surface of the receptor. Drugs that are antagonists can be used to treat a drug overdose. For example, naloxone, which binds the same mu-opioid receptor as morphine. Now let's talk about receptors. A receptor can be defined loosely as a molecule that recognizes specifically a second molecule whose binding brings about the regulation of a cellular process. In the unbound state, a receptor is functionally silent. Receptors are tightly regulated by protein structures that bind specifically to a particular ligand. In reality, a specific ligand can activate more than one signaling pathway via different receptors. Membrane-bound receptors can initiate responses from one or more ligands through their binding sites on their extracellular domains, and can initiate signaling transduction in their intracellular domains. Cytosolic receptors can bind hormones and become active, allowing them to alter signaling pathways in the cytosol or enter the nucleus and alter transcription. Xenobiotics can discriminate between receptor types. Small modifications of ligands can result in attenuated or enhanced cellular response when binding the same receptor. Drug developers take advantage of this and chemically modify drugs to increase their specificity and decrease their adverse effects. Receptor types arise from multi-gene families and can vary depending on the tissue or organ. The body can also control cellular responses by up or down regulating certain receptor subtypes and associated proteins in specific cells, resulting in various tissues having different responses to the same ligand. It is important to keep this in mind as we talk about the body's reaction to a drug or toxin and how this can lead to a therapeutic or toxic response. There are many different types of receptors in the body, including G-protein coupled receptors, receptor tyrosine kinases, ion channels, nuclear receptors, and enzymes. Take a look at the provided videos on G-protein coupled receptors, receptor tyrosine kinases, and ion channels to familiarize yourself with signaling pathways for each receptor. Nuclear receptors are critical for hormone signaling, while enzymes like COX-1 and COX-2 are major targets for NSAIDs, or non-steroid anti-inflammatory drugs like aspirin.